Welcome to practical number 2 under food chemistry course module. Determination of the effectiveness of heat, ion concentration and pH on functional properties of protein. I am Yasara Kodaguda, your practical in charge demonstrator. And I hope you have already gone through the practical handout before joining to the practical lesson. Under the practical title number 2, our subtopic is determination of water holding capacity of meat. So you have already learned in the lectures that protein has number of functional properties. As well as protein plays a major role in sensory attributes like mouthfeel, texture in many foods. So in this practical class, we are going to talk about water holding capacity of proteins in meat. You know, in meat, it basically contains protein in addition to water. Well, what is water holding capacity? Water holding capacity is the ability of protein to retain water. That can be its own inherent water or added water. It affects sensory attributes of meat like visual acceptability, juiciness, tenderness, weight loss and cooking yield of meat. Right. You know that protein molecule has a globular structure or the three-dimensional structure. In this structure, protein molecules, a hydrophobic part of the protein molecules are oriented inside the protein molecule. It's called core of the protein molecule, while the hydrophilic parts are aligned in the surface of the protein molecule. This is how the protein molecule is present in aqueous solution. Because of the repulsive force of the hydrophobic parts with the water molecule, they hide inside the protein molecule. This picture illustrates how protein molecules bind with water inside the cell in meat using hydrogen bonds. Now let's see how to determine the water holding capacity. For that you may need 3 grams of finely ground meat or whatever the protein source that you need to measure the water holding capacity. First you have to measure 3 grams of protein source into a pre-weighted centrifuge tube. Then add 15 milliliter of distilled water into it. After that, vortex the mixture for 10 seconds every 5 minutes for total 30 minutes of holding time. Make sure whether the material is wetted thoroughly. After that, centrifuge the mixture for 10 minutes at 2000 rpm. Once complete, decant the supernatant and discard it, and record the weight of the sediment with centrifuge tube. You can watch the video on practical demonstration of the test using the given link below. In order to calculate the water holding capacity, you have to subtract the final weight from the initial weight. Water holding capacity can be expressed as gram of water retained in 1 gram of protein. You can try activity number 1 and do the calculations and submit it after the practical lesson. Because of the repulsive force of the hydrophobic parts with the water molecule, they hide inside the protein molecule. Right. You know that protein molecule has a globular structure or the three-dimensional structure. In this structure, protein molecules, a hydrophobic part of the protein molecules are oriented inside the protein molecule. It's called core of the protein molecule. At isoelectric point, protein cannot bind with water. due to lack of net positive or negative charges. Well, now let's see how to determine the water holding capacity at different pH conditions in order to find out the effect of pH. First, get the empty weight of 6 centrifuge tubes and weigh 3 grams of finely ground meat into each tubes. Next, add 15 milliliters of distilled water into them. Measure the pH of each tube after vortex them. Then, adjust the pH of each mixtures into given pH values to make a series. After keeping them for 10 minutes, check the pH of the tubes and get it to the required value. Once complete, centrifuge each tubes for 10 minutes at 2000 times G. After the centrifuge, carefully decant the supernatant and discard the water and get the final weight of each centrifuge tubes. Finally, calculate the water holding capacity of meat at different pH condition using the equation. Now you can plot a graph for water holding capacity against different pH. When the water holding capacity is plotted against the pH of the media, you can obtain this kind of a graph. pH at minimum water holding capacity denote the isoelectric point of particular protein. That's why protein precipitates at its isoelectric point. Increasing or decreasing the pH on either side of isoelectric point will result in an increased water holding capacity by creating a charge imbalance. After completion of the practical, you have to do the activity number 2. You have to describe the effect of pH have on the weight of sediment muscle and how you can relate the hydration of muscle protein to this. Explain and illustrate the chemical changes responsible for these effects. Locate the isoelectric point of myofibrillar proteins in the graph plotted water holding capacity against pH. Well, this is the end of our practical class. I hope you have gained much knowledge about determination of water holding capacity, and you are supposed to complete a performer given for this practical. And thank you so much for your cooperation.